DNA replication, an important process for all life functioning. Without it, we couldn't grow or replace or repair damaged cells. This lesson is on the DNA replication or DNA synthesis. So, DNA has the ability to replicate itself, and this is necessary to maintain a consistent level of genetic material through each level stage of mitosis. And so each cell is now able to divide itself into two cells that has identical genetic information. And the replication takes place during the S phase of interphase of the cell cycle. So replication of DNA is semi-conservative. So each new molecule of DNA is going to contain one original strand of DNA and one new complementary strand to the parent. So now each double helix conserves half of the original molecule. So when we make a new double helix strand, there's one original and one new strand of DNA. And this takes place at several locations along the DNA model simultaneously. But the steps are described in sequence so that we better understand the concept. So this is happening all the time throughout the DNA and at different speeds. So replication starts at a specific nucleotide sequence called the replication origin. And during replication, the weak hydrogen bonds that hold together the nitrogenous bases are broken. And this happens with a group of enzymes called helicases. So we're unzipping the helix. So helicases are the enzymes that unzip the DNA or break apart those weak hydrogen bonds. This is going to create two Y-shaped areas known as replication forks at the end of the unwound area. And this unwound area is going to be called the replication bubble. Now, the original parent strands are conserved and they maintain their integrity, while two new strands form within them. So those two parent strands act as a template, and complementary DNA bases are added on either side of the replication fork. So free-floating nucleotides that we maintain in our cell from our diet are attached to the exposed nitrogen bases according to the base pair rule. So adenine bonds with thymine and cytosine bonds with guanine using DNA polymerase. And the process of adding new nucleotides is known as elongation. So we're elongating our DNA. The DNA polymerases attach new nucleotides from the 3' end to the 5' end. And elongation can only take place in a 5' to 3' direction. But this is information from IB, so this is just information for you. It's nothing that you need to have for your diploma exam. So replication occurs in opposite directions along each strand of the DNA. So it will go 5' to 3' in this direction and 5' to 3' in this direction. One strand is able to be replicated continuously, so they go in order, and this is known as the leading strand. The other strand is replicated in short segments, and this is known as the lagging strand. And those short segments are called Okazaki fragments, and they are going to be held together by another enzyme called DNA ligase. So DNA helicase is going to be the enzyme that unzips our DNA, breaks apart those hydrogen bonds. DNA polymerase attaches the new nucleotides together. And the DNA ligase attaches the short segments called Okazaki fragments back together on the lagging strand of replication. So since DNA polymerase cannot synthesize new fragments of DNA, and the RNA primer serves as a starting point for elongation, an enzyme called primase is required to construct as a primer. So then the DNA polymerase removes the primer by eliminating nucleotides. So it kind of goes and then cleans everything up at the end. Hydrogen bonds form between the nitrogen bases, and then there's proofreading enzymes that check the new DNA strand for mistakes. Errors are going to be removed by cutting mistakes out and using an endonuclease and replacing the correct nitrogenous bases. So during DNA replication, our cells go through a lot of effort to make sure there's not errors in DNA replication. So as soon as the new strands are formed, they rewind automatically into the double helix structure, and replication continues until the new strands are complete and the DNA molecules are going to separate. And this is going to be called termination. So this replication produces two strands of DNA from one, where each strand is going to be half new and half old, known as conservation. So here we can see the DNA polymerase, and that is going to be binding the nucleotides together, and the yellow DNA ligase, which is holding those small Okazaki fragments together on the lagging strand. So in this model at the top, we have helicase as kind of like that yellow egg-shaped oval that is unzipping the hydrogen bonds. We have the leading strand on the left that is replicating the 5' to 3' end using DNA polymerase to attach those nucleotides together. On the right-hand side, we also have a ligase that is going to be holding the Okazaki fragments together so that they can be joined successfully. Now, as humans and scientists, we use the DNA replication process and the enzymes to our advantage in certain situations. So, one of them is genetic engineering and recombinant DNA. So, genetic engineering refers to the alteration of an organism's genome by selectively removing, adding, or modifying DNA. And we have done this successfully in a variety of different plant food organisms. Recombinant DNA occurs when the process of cutting out DNA from one genome and placing the DNA into another genome, resulting in a transgenic organism. And so we do this to modify bacteria for medicine and bioremediation, so environmental cleanup. And transgenic plants are used to improve crop yield and resistance to environmental effects, like Roundup Ready canola, where you can spray Roundup on your crop. It will kill the weeds, but your plants won't die. Also, this has happened occasionally for cloned livestock and cloned organs for human use. So when we want to cut the DNA, we need to use something known as a restriction enzyme. So that restriction enzyme is going to cut DNA at specific sequences in the DNA strands. And usually these are DNA strands with repeating letters. Then the ligases are going to fuse those Okazaki fragments together. Scientists use those as a biological glue to insert new fragments of DNA into the original DNA strand. So we use restriction enzymes to cut DNA at specific sites, and then we use ligase, an enzyme, to fuse the segments of DNA to insert new genes onto the original strand. So we're trying to use gene therapy to help people with diabetes produce their own insulin. So a gene in the human genome that codes for insulin is cut out using restriction enzymes from an individual with normal DNA. The plasmid of an E. coli bacteria is then going to be cut using a similar restriction enzyme so that the gene from insulin can be inserted. And we use a similar enzyme so that it cuts the gene at the same location, allowing for the ligase to more easily glue those two gene sequences back together. The bacteria can read the human DNA, and then it produces insulin for us to use and harvest later on. And this process is also being used to then possibly insert those genes into the pancreas of somebody who has diabetes so their body can produce its own insulin. So another example for the insertion of gene is a gene that codes for human growth hormone into animals so that they grow faster. But lots of these biotechnology developments have a lot of ethical issues that people consider before they're approved for use. So what are some of these issues? The cost of this, is this an effective place for money to be spent by corporations and governments? Is there motivation for this product? Do people want to eat livestock that have had growth hormone introduced into them? The biological characteristics of the product. Are we going to have a whole bunch of really large cattle now that aren't going to fit into typical cattle liners and slaughterhouses? The environmental effects. Who owns this technology and the patents? And also, it's important when we're looking at gene therapy for individuals, is there freedom of information and privacy concerns? And can people own the DNA of other individuals? So restriction enzymes are also used when it comes to gel electrophoresis, which is a technique that's used to separate DNA fragments for the purpose of identification in like crime scenes or paternity tests. So a sample of DNA is taken from an individual and compared to either a child, siblings, and or a crime scene sample. And so this produces then a number of DNA segments of different lengths. And those different pieces of DNA are then tagged with a radioactive isotope. And some of the pieces of DNA are short and some of the pieces of DNA are long, depending on which individual they came from. Since my sample and anyone's sample would be cut with the same restriction enzymes, but because our genes are slightly different, they're going to be slightly different lengths. So then we're going to use an agarose gel that contains wells along one side, and the samples of DNA are inserted into those wells. So they would take a known sample, say like from a crime scene, and then they would take their 6 suspects that they're going to compare with it. Then an electric current is going to be run through the gel, causing the movement of those DNA fragments to move up the agarose gel. The shortest strands are going to move the farthest because they have the lowest weight, and the longest strands, because they're heavier, will not move as far. So imagine you have a bunch of small pieces of paper in your hand, but they're all different sizes, and you blew on them gently. The lighter ones would travel farther, and the shorter ones would travel, the heavier ones would travel a shorter distance. So then this is going to separate the DNA into its different bands. And once the gel is left to set out, you're able to stain them with an X-ray film and compare the samples with one another. So we can see here that we have different lengths of DNA cut, and it depends on what you're comparing it to. But we can see it looks like samples, the first 2 samples and the last 2 samples are very similar in where those DNA bands have been left. So DNA fingerprinting was developed in 1985 to determine whether or not a sample of DNA came from a specific individual. People generally have similar DNA, but every person, with the exception of identical twins or triplets, has some unique non-coding segments of DNA. Called introns, and exons are segments of DNA that code for proteins, and basically humans have all the same coding for proteins because we all have the same life functions. So when that sample is placed through gel electrophoresis, we can compare the samples with each other, and we have a different number of sites where those restriction enzymes are cutting our DNA, so we have a different amount of long and short segments. So here in lane A, we have crime scene DNA cut with enzyme 1, and in lane B, crime scene sample cut with enzyme 2. So we want to go and see what matches. And if we go through column C and D from suspect 1, we can see they don't match. But if we go through column E and F from suspect 2, we can see that they do match. So this is just one of the uses of gel electrophoresis and DNA technology. I hope you enjoyed today's lesson. See you next time.